In the traditional times, biotechnology literally started from the brewing industry. Now, what is brewing? Brewing is the preparation of beer. Now, you take some cereals, the cereals contain starch, you convert that starch into sugar using chemical processes, and then you introduce some microorganisms called yeast and give it a proper environment, and those yeast cells will ferment this sugar into alcohol. Now, what is this word ferment? Fermentation is the process of conversion of one set of products, usually sugar and similar substances, into alcohol, carbon dioxide, and other useful products. In this case, you take the cereals, you convert it to sugar, and then you keep it in this nutrient medium of yeast for a certain number of days, and you get alcohol. This alcohol is nothing but beer. Similarly, instead of cereals, if you take fruit, then you get wine, and you're not using the carbon dioxide or the other useful products, you're using the alcohol. So this beer or wine that you get usually contain 10 to 20% alcohol, and therefore they are called soft liquor. But you can distill this soft liquor into hard liquor. This is what happens. Distillation is the process of physical separation of the constituents of a mixture. So just consider an example. A plus B plus C is a mixture of three substances A, B and C, each having different boiling points. So when you boil, say A boils first, you collect its vapors in a certain container, then B boils, you collect its vapors in another container, and C in another container. That's how you separate this mixture. Then you can further purify A into A1, A2 and A3. You can keep purifying till the alcohol concentration increases, and therefore when you do distillation, so distillation is not a chemical or biological process, it's completely a physical process. So when you distill soft liquor, the alcohol concentration might increase to more than 40%, 50%, 60%, 70%. Then you get what is called hard liquor, such as gin, rum, whiskey, and vodka. There's a hard liquor called absinthe, popular in Europe, and that contains easily more than 60 or even 70% alcohol. So this is about conversion of the starchy substance into alcohol using a process called fermentation. Literally, the yeast is consuming the sugar and excreting alcohol. So you are eating the excretion of yeast, if you think it that way, but don't think it that way. But just imagine how useful is this organism called yeast, which is consuming sugar and producing alcohol for you. But it is also producing carbon dioxide. So if you choose to disregard the alcohol portion and utilize the carbon dioxide, then you can leaven a loaf of dough and you can create bread. That is how bread is produced. And you can also use other microorganisms such as lactobacillus instead of yeast, and you can produce yogurt and cheese, again through the same process of fermentation. Again, alcohol is not just used for consumption. Of course, the alcohol which is being produced here is ethanol, but it is not just used for consumption. It can also be used as a biofuel. In fact, there is a program for blending of ethanol in gasoline to the tune of 10 to 20%, and with the evolution of car engines, this percentage might increase. So we can literally produce vehicle fuel not from the fossil fuel reserves in the earth, but from crops, from cereals, from sugarcane. And that is why the sugar mills are finding profitability these days, because they are not just refining sugarcane into sugar, they are also having distilleries. They are producing alcohol for consumption, they are producing ethanol for fossil fuel blending, and there's a whole lot of possibilities. And biogas can also be produced using the same process of fermentation. So fermentation is that important. That was literally the traditional beginnings of biotechnology. But fermentation has many more possibilities, and that is antibiotics. So if I take certain bacteria or fungus, put it in a nutrient medium, and then do the fermentation process, what happens? I already took yeast, I already took lactobacillus, and they produced alcohol, they produced carbon dioxide, and I utilized those products. But there are certain fungi and bacteria which on fermentation in proper nutrient medium produce certain chemicals called antibiotics, which kills other bacteria. These antibiotics can kill specific bacteria or they can kill bacteria in general. But they are so important because human beings are infected with bacterial diseases. So if we can use these antibiotics to kill the bacteria that is multiplying within our body, then we can treat these bacterial diseases. So the first antibiotic was discovered in 1928 by Alexander Fleming. He took the fungus Penicillium notatum and fermented it for a few days in a petri dish and produced this antibiotic called penicillin. And that was useful in treating a wide array of bacterial diseases in humans, but that wasn't tested until the 1940s. Again, there is a bacteria called streptomyces. When you ferment that in a petri dish, you produce an antibiotic called streptomycin, which can actually kill another bacteria called Mycobacterium tuberculosis, which causes tuberculosis in human beings. So the fermented antibiotic produced from one bacteria, streptomyces, can kill another bacteria, Mycobacterium, and can act as an antibiotic. That is the utility. Thousands and thousands of antibiotics have been produced in this way, and it is extremely useful. So when a body has a lot of these bacteria which have multiplied and are causing disease in us, we can consume antibiotics and they can act as effective medicines. But what if I want to augment this process? What if these antibiotics are not very effective? What if the bacteria has multiplied in my body too much? So in that case, I need to understand how the human immune system works. The human immune system produces certain compounds, certain chemicals called antibodies or immunoglobulins, which detect an antigen or a foreign toxin inside the human body, attack and destroy it. And that is naturally done by the human body. So, whereas antibiotics act artificially, externally, these antibodies are produced naturally inside the human body. But when we are infected with tuberculosis bacteria or diphtheria or tetanus, and the bacteria multiplies very quickly, then the body might not be able to produce antibodies at that rate. In that case, we might require to inject antibodies externally. So what is done? Say for example, a lot of bacteria has already multiplied in a patient. He needs immediate treatment. So you take a horse, you inject it with a certain dose of that particular bacteria, and within a few days, the horse produces a good amount of antibodies in its blood. You take out a sample of the blood of the horse, you remove the platelets, you get the serum, you further remove the RBCs and WBCs, and you get the liquid part called plasma, which is rich in antibodies. You store it, and whenever such a patient comes to the hospital, you give it a dose of these antibodies. By the way, they are called equine or animal antitoxins. They are a cocktail of antibodies produced within that horse, or for that matter, it can be a sheep or a goat. And when it is injected in that patient is treated because these antibodies kill the bacteria very quickly, so they can act as supplements to the antibiotics. But there are two issues. First of all, these antibodies in the plasma of the horse are a cocktail of antibodies. They might not be specific to that particular bacteria which is infecting the human patient. And that's why they are called polyclonal antibodies, because the horse blood will produce a lot of, for that matter, even if instead of horse you take a human being, you get a human derived antitoxin. And that also is a cocktail of antibodies. So this cocktail of antibodies is called polyclonal antibodies, and they are produced in vivo and usually by exposing an animal to an antigen. But there are certain issues. For example, if you take the antitoxin cocktail from a horse, then of course it is an animal product and you are injecting it into a human patient. There can be chances of rejection, chances of allergy, and of course it has lots of antibodies. It is not just specific to that particular disease. So we turned to human-derived antitoxins, where instead of a horse, we take a human being. And a human being who has recovered from that disease can donate his plasma to a patient who is presently suffering from that disease. This is exactly what was tried during the coronavirus pandemic. When a patient went into advanced stages of symptoms, then his body was unable to produce that many antibodies, then the plasma of recovered patients was used. But they are all polyclonal antibodies. And if you go for the natural way, then you will get polyclonal antibodies. So I want the specific antibody against coronavirus or against tuberculosis or against whichever disease I'm treating in the patient. So that specific antibody is called monoclonal antibody. So just for the perspective of it, antibodies in the human being is produced by immune cells called the B cells. They are also a type of white blood cells. These B cells produce antibodies. So there are certain specific B cells which will produce specific antibodies to an antigen. If I extract those specific B cells, they will produce the antibodies for that particular disease. But not many B cells like that exist in the human body. And that's why we are not able to produce monoclonal antibodies naturally within our cells. Maybe a million years down the line, after a lot of evolution, we might be able to naturally produce monoclonal antibodies against tetanus or tuberculosis or coronavirus. But right now, it's polyclonal antibodies which are produced naturally. So these specific B cells are pretty rare in the human body. So I take it out. I want it to multiply enough continuously, sustainably, and produce enough monoclonal antibodies. So in order to do that, a technology called the hybridoma technology is used, which was invented in the 1970s, where this specific B cell is fused with cancerous B cell or myeloma cell, the purpose of which is to create an immortal hybrid cell line capable of infinitely producing a single highly specific antibody. And in this way, within the petri dish, I produce sufficient quantity of monoclonal antibody which I can inject into that patient. And this treatment has been successfully used against a lot of diseases, especially in advanced symptoms, patients exhibiting advanced symptoms. But all of these things are when the disease has advanced too much into the human body. Why not arrest the disease at the beginning? Why not nip it at the bud? That is the very concept of vaccines. The first vaccine was discovered against smallpox by Edward Jenner. And the basic concept is this: whenever your body is exposed to an antigen, to a virus or bacteria or anything, it produces these antibodies. Why not expose your body to very mild doses of this antigen and let your body produce the antibodies so that it's all very much under control? And then in the future, 10 or 20 years down the line, when in case you get contracted with that disease, in case that antigen attacks you, immediately the swarm of already existing antibodies in your blood will be able to kill that antigen at the very onset. It will not be able to multiply inside your body, and it will be killed even before you get the symptoms or before any damage is done. In other words, it will be nipped in the bud. That is the basic concept of vaccines. So for that, I need to produce some controlled doses of this antigen, whether virus or bacteria, and that I can do it in a petri dish. So the concept used here is cell culture. I talked about fermentation, where the basic intention of growing these microorganisms in the petri dish was to produce a product of our interest. But here the intention is not to create any product of interest. Here the intention is to just multiply the cells of the virus or the bacteria in a petri dish to a sufficient quantity, and then you kill all these cells, or you inactivate them, or you attenuate them, so that when injected in the human body as vaccines, they don't cause any disease. They just trigger an immune response which creates an adaptive immunity inside the human body. And ever since the smallpox vaccine was discovered, vaccines for so many diseases are discovered, and vaccination is the priority treatment. I mean, prevention is better than cure. And it's only when the vaccine for coronavirus was discovered that the pandemic really stopped or started slowing down. The curve started flattening. The curve would have flattened anyway, that is epidemiology, but it would have infected so many human beings, more than 60 to 70% of the human population on Earth before the curve would have flattened. And so many deaths would have occurred in patients who couldn't have sustained the onslaught of the symptoms. So, vaccination prevented that and flattened the curve initially. So, that is the importance of vaccination in preventing epidemics and in preventing individual disease from happening at the first place. That's why we have routine immunization programs where children, right after they are born, are given different vaccines in different age groups. So, this was about vaccination. And this brings me to another exciting topic called stem cell culture. Stem cell culture, I've already talked about the cells of the antigen being cultured to produce a vaccine, but stem cell culture is beyond that. What are stem cells? Are they cells of the stem of plants? No. They are cells inside the human body which, unlike other cells which can only divide into their own types, a skin cell makes a skin cell, a bone cell makes a bone cell, a muscle cell makes a muscle cell, but a stem cell can make different types of cells. It can make a bone cell, it can make a blood cell, it can make a adipose fat tissue cell, depending on the requirement. And therefore, stem cell culture is important, especially in treating various diseases. For example, if a person has a disease where defective red blood cells are formed, so in that case, I need to understand first of all that red blood cells are formed from hematopoietic stem cells in the bone marrow. So I take the healthy hematopoietic stem cells from a healthy donor and put it in a stem cell culture for a few days, maybe for a few hours, depends. Let it multiply, let it grow, let the pH be all right, and then I inject it into this patient, and then the patient gets this cocktail of healthy stem cells which produce healthy RBCs, and his disease is cured. A lot of cancer is cured in this way when things go bad. A lot of genetic diseases are cured in this way. I'll just give you an example of sickle cell anemia. The sickle cell anemia is a disease which infects a lot of tribals and Africans, and that's one of the most common inherited genetic diseases in the world, and it is caused when a particular gene, in fact a particular nucleotide base in a particular gene has gone wrong, and that's why the hematopoietic stem cells produce defective hemoglobin, and hemoglobin is the majority of the red blood cells, which is why the red blood cells, instead of being donut shaped, become sickle shaped, and they get stuck in the blood vessels. They can create a lot of pain, a lot of problem, maybe stroke, heart attack, even death in the later stages. So it is a pretty serious disease and needs to be cured. So the cells, the hematopoietic stem cells, are producing this defective hemoglobin, the defective RBCs. So you take a healthy donor, a donor who is closely related to the patient, who has healthy hematopoietic stem cells, and you take a cocktail of that stem cells, you culture it in a cell culture, and then you inject it into this patient. When the patient gets a dose of healthy stem cells, he will produce healthy RBCs. Similarly, when a person suffers from cancer, especially advanced stages of cancer, then you use chemotherapy. In fact, you also need to use chemotherapy in the case of the sickle cell disease, because you want to kill the defective stem cells before injecting the healthy ones. So in the case of cancer, you do chemotherapy, you kill the defective stem cells, and then you inject this dose of healthy stem cells from a donor. The problem in this is that you require a donor. And by the way, this process of stem cell transplant is called allogenic stem cell transplant, and it requires a donor. And even when the donor is closely related, there is a possibility of rejection, allergy, and whatnot in the patient. And of course, chemotherapy is required. So why not take the stem cells from the same patient, and if they are healthy stem cells, if you take it from one part of the body, they are healthy, and then if you can re-inject it back to the same person, then there will be less chances of rejection. But what if all the stem cells in that person are defective, they have that defective gene? In that case, you need to take the stem cell from that person and you need to modify the gene. You need to modify this gene and then re-inject it back to the patient. Well, this is the process of genetic engineering. So by modifying a gene, I mean you might need to add a certain gene, you might need to delete a certain gene or replace a certain gene. So let me begin with adding a certain gene. So consider this as the DNA. It contains gene number 1, 2, 3 up to 100. I need to add a 101th gene in this DNA fragment which has some desired characteristics. So for that I use a technology called the Recombinant DNA Technology. So this is how it goes. First of all, I need to understand where is this DNA fragment lying. Of course it's lying inside the nucleus. Where is the nucleus lying? Of course it's lying inside the cell of the patient or whichever organism. And the cell has a cell membrane or cell wall. So first of all you need to insert this 101th piece of gene into the cell, then it has to go into the nucleus and then it has to merge with the DNA. How do I introduce this gene of interest or this insert DNA fragment? Well, I need to isolate it from a particular organism. Say for example, I'm giving a very wild example. Say for example, I want to insert the gene of a horse inside a donkey. These are pretty wild examples. But say if I want to do that, first I need to isolate the gene of the horse. And then using a vehicle or a carrier, insert it into the cell of a donkey. Let me give you a real life example. There's a bacteria called Bacillus thuringiensis which has a particular gene which produces a chemical which kills the bollworms which infect cotton plants. So, I mean, the bacillus doesn't need that chemical, the cotton plants need that chemical. So I want to take this gene and I want to insert it into the cotton plant. So this is what I do. This is the bacterial cell. I dissolve the cell wall, cell membrane, I dissolve the nuclear membrane, and I isolate the DNA molecule. Now I treat it with restriction endonuclease enzyme which cuts the DNA into separate pieces. I separate all of these pieces using electrophoresis and southern blotting techniques, and then I get my gene of interest. By the way, this has sticky ends. This is a single-stranded DNA. It's a double-stranded DNA, but the sticky ends are single-stranded, so they are ready to be attached with something else. This is my gene of interest alpha. Now I need a carrier. Just as we want to go from Delhi to Mumbai, I can't go on foot. I need a bus or a car or some vehicle, alright? So similarly, this gene of interest or insert DNA cannot be inserted into the cotton plant as it is. I need some vehicle. So that vehicle can either be a plasmid, which is a piece of DNA inside a bacteria. That acts as an excellent vehicle. It can also be an adeno-associated virus, which has been time tested to act as such a vehicle. Let me consider plasmid for this example. So I similarly take a bacteria and I dissolve its cell wall. I take out the plasmid. I cut it using restriction endonuclease enzyme, and again it has a sticky end. This is the vector DNA or the carrier DNA. Let it be called beta. I add alpha and beta. I ligate them using the DNA ligase enzyme, and I create a chimeric or recombinant or hybrid DNA. Let it be called gamma. This gamma is a vehicle having my gene of interest, and I am ready to put it in the host cell. So how do I put this gamma piece of DNA into the cotton plant? First I put it into a bacteria called agrobacterium, and then I use this agrobacterium to infect the cotton plant so that it gets absorbed into the genome of the cotton plant. Then I isolate the seeds of that cotton plant and sell it in the market as GM cotton. This process is called agrobacterium mediated transformation of the cotton plant. By the way, how did I insert this gene into the agrobacterium? I used a gene gun in a process called biolistics to insert this gene into the bacteria. So this entire process, first I transformed the bacteria, then I transformed the cotton plant. This process is called transformation. And then the cotton plant will start expressing that gene. It will start producing that desired product, that toxin which kills the bollworms, and we do not require any pesticides externally. It is such an advantageous method, and it has been widely used right now. So this is about adding a certain gene of interest, but it's very, 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 very controversial if you want to add a certain gene in a human being belonging to some other organism. It has not been tested, and it should be very cautious when even considering such propositions, because it's highly ethically controversial. But there are situations where you may have to delete a particular defective gene in a human body. Let me again go back to the example of sickle cell anemia. As I said, that this disease is caused by defective hemoglobin being produced. Now, when we are in the uterus of mother as fetus, we produce fetal hemoglobin. As soon as we take birth, there is a certain gene which deactivates the production of fetal hemoglobin, and adult hemoglobin starts to be produced. And in case the person suffers from sickle cell anemia, he will produce defective hemoglobin as an adult. So if we can delete this gene which produces the defective, if we can delete this gene which deactivates fetal hemoglobin at birth, we will be able to sustain the production of fetal hemoglobin even as adults. So we require deletion. In order to delete, we need to cut this piece of DNA, and the cutting of this piece of DNA initially happened using technologies such as zinc finger nucleases and talons, but ever since 1990s it has been happening since the CRISPR-Cas9 technology. So let me talk about it. In 1990s, scientists found that in about 40% of bacteria, there seemed to be clusters of regularly interspaced repeating sequences called clustered regularly interspaced short palindromic repeats, because they were palindromic, or CRISPR. So just consider this as a fragment of DNA, and it has these CRISPR patterns which are palindromic. Let it be GAG, 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 and so on. In between them, you had spacer portions which were all unique, and scientists couldn't figure out what they meant until a scientist found out that these unique spacers resembled viral genome of different viruses. And then it clicked that yes, it might act as the immune system of bacteria. So whenever a bacteria is attacked by a virus or a bacteriophage, then the immune system identifies a particular portion of the viral genome, and it takes that viral genome and adds it to this CRISPR spacer pattern. So this CRISPR array acts as a sort of memory book for the bacteria. So whenever a virus attacks, having a viral genome very similar to one of these spacers, then there is a certain enzyme within the bacteria called the CRISPR associated enzyme, or Cas9, which identifies which has this guide RNA. So let me bring it to perspective. DNA is transcribed into RNA. Everybody knows that. So these portions of spacers will of course be transcribed into corresponding RNA. This corresponding RNA gets associated with the corresponding Cas9 enzyme which is present inside the bacteria, and this bacteria along with the guide RNA keeps traveling until it discovers that a virus has invaded which has a similar genomic sequence similar to the guide RNA. And how does it identify it? It identifies it using something called a protospacer adjacent motif, or PAM, in the viral genome. So the portion of the viral genome which is genetically similar to the spacer is known as prospacer, and you will find this PAM motif adjacent to the prospacer having a pattern of NGG. So two guanines, and N can be anything, it can be A, T, C, or G. So whenever it finds this PAM pattern in a virus, it looks whether the guide RNA matches with the protospacer sequence, and if it matches, that's it. It goes and cuts it and destroys the virus, and the virus is unable to infect this bacteria anymore. So scientists found out that, wow, it is this cutting of the virus by the Cas9 enzyme using this guide RNA which creates immunity. Why not utilize this Cas9 enzyme for cutting a piece of DNA of our interest, and we'll let the Cas9 enzyme know about it using the guide RNA, and it will help us to cut any piece of human DNA as well. Why not cut that gene in the human DNA which deactivates fetal hemoglobin at birth? In that case, fetal hemoglobin will be continue to be produced even as adults, and in that case it will mask the defective adult hemoglobin, and symptoms of sickle cell anemia would not appear in the patient. That's exactly what they did. They fed the guide RNA similar to the gene of interest which deactivated fetal hemoglobin, and the Cas9 enzyme cut out that gene, deleted that gene. And this technology, this therapy is being used in a lot of places right now in the world. There is another technology. So I've talked about deletion using the CRISPR-Cas9 technology. There is another technology called gene replacement. Now, whenever you delete a gene, the natural DNA repair mechanisms in the body will kick in. And it is utilizing these mechanisms that you can insert your gene of interest in. And how will you introduce that gene of interest into the cell? Using that same vector recombinant DNA technology. So in that way, you will be able to literally delete a bad gene and insert a good gene. So there is this bad gene which produces defective adult hemoglobin. You can replace it with a good gene. So without involving any fetal hemoglobin, which might have side effects, because fetal hemoglobin does have side effects that are also being studied. So without messing with that, you can literally replace the defective sickle cell gene, and that's it. So that's how you can add, delete, and replace genes. I'm not using the word gene editing. A lot of textbooks use this word because I am literally replacing. However, if I go and chemically alter the nucleotide bases to edit that gene, then it is called gene editing. And this base editing is also a technology which is happening. So this is how you can do. And when I discussed about deletion is one of the most important things to treat a lot of genetic inherited diseases in human beings. But even without actually deleting a gene, we can achieve the same objective using a technology called RNAi, or RNA interference. So when let us not delete that gene, all right? Let us not delete the defective gene. Let it get transcribed to RNA. But when that RNA is being translated to protein, then we do not allow that particular portion of the RNA to be transcribed into protein. How we use a piece of DNA called a piece of RNA called siRNA, small interfering RNA, which masks or blocks that particular portion of the genetic code, and that is not translated into a protein. And this is exactly how we can prevent the defective protein from being expressed even without deleting that gene from the genome. And this RNAi technology is also being studied and used big time. All right, so this is the latest genetic engineering things which are happening, and this must excite you about biotechnology. But there are many more things happening. And let me go to biosensors. Now, we know that in electronics there are two types of transistors: the bipolar junction transistor, which depends on the base current, and that changes the output current. The base current acts as the input current, and that changes the output current. Similarly, we have field effect transistors in which the gate voltage, any slight change in the gate voltage can change the source and the drain currents. This change in the gate voltage can happen because of certain biomolecules, even because of some force. So it can be used to measure certain force in our muscles. It can be used to detect the presence of certain biomolecules and help in pathological tests. It can be used in immunoassays, in which a lot of pathological tests can be done using this thing. And also, glucose sensors have been made using this very working principle. Moreover, DNA can also store information. It can be used in computational applications, and that's the realm of bioinformatics. Another thing is the environmental role of biotechnology. There is a bacteria called Pseudomonas, which naturally breaks down oil in oil spills. And a lot of genetically modified organisms can also be used to treat metal pollutants and solve pollution big time. They can be used in biofiltration, biodegradation, bioleaching, and so on. I just want to end this discussion with something which might be a darker picture of biotechnology, and that is eugenics. Eugenics is a highly controversial aspect, and that is modifying the genome of a human being to incorporate certain chosen traits and features. For example, if you want a human being to have brown hair, if you want a human being to have blue eyes, then you genetically modify. Now, you're playing with God. God has made you one way and you want to become another way. And this is controversial because, first of all, it will reduce human diversity, and secondly, you can change anybody. It is highly ethically controversial, and it is coming up as we move into the future. But the technology is not that new. The technology is pretty old, thousands of years old, and we have been using the same technology of selective breeding in plants. So what we have been doing in plants, it is not really biotechnology. I mean, selective breeding in plants have happened for thousands of years, ever since the Neolithic Revolution developed agriculture. We've been taking favorable breeds and we've been rejecting the unfavorable breeds. Of course, this has reduced the diversity of the different breeds. Now we use almost very similar breeds of rice and wheat throughout India. Throughout the world, there are only a few varieties of rice or wheat, because the traditional varieties, I think they are called land races, they are not being entertained. I mean, the rice variety which will be useful in Bengal, and the rice variety which will be useful in Kerala, and the rice variety which will be useful in Punjab, and the rice variety which will be useful in Vietnam and will be useful in China, they will all be different depending on the weather, the soil, the water conditions, everything. But because of the selective breeding, we are trying to use the same varieties, and that is sure to reduce the diversity, that will not be able to adapt properly in the right environment. In some places the yield will be more, in some places the yield will be less. If a pest attacks, if a disease attacks, it will wipe out the entire field at once. So all these problems will keep happening. Susceptibility to not just diseases but even to changing environment increases. So let us look at the positive side of this. We can do genetic screening of individuals at birth. Even before a child is born, we can understand what are the diseases that the child can develop genetically, and we can nip it in the bud. And we can also give personalized medicine. These are all applications of biotechnology. I hope that by covering the wide variety of applications, starting from brewing and biofuels to antibiotics to antibodies to vaccines to stem cell culture to genetic engineering to biosensors, you have understood some portions of the exciting world of biotechnology. I just want to end with one technology called cloning. Now, we usually use the word cell culture for animals. For plants, we use the word tissue culture. And why is it so? Because we use whole pieces of tissue, pieces of their leaves or stems, in the case of rose plant, for example, to create whole new plants. For example, we can take a stalk of the rose plant with one bud and grow the entire rose plant in a petri dish. If you do the same thing in the soil, that is vegetative propagation. In the petri dish, it's called micropropagation. So this is tissue culture. What if I want to create a whole new human being in a petri dish? Is it possible? That process is called cloning. So what I do, if I want to create the clone of person A, I take his somatic cell, maybe skin cell or something, and I remove the diploid nucleus and I keep it. Then of course, if a clone has to be born, a mother is required. So I have a surrogate mother. I take her egg cell. Egg cell is haploid, waiting for sperm cell to fertilize it and make it diploid, but that never happens. You remove that haploid nucleus and you inject the diploid somatic cell nucleus of the person A, and you re-inject it back into the uterus. And you're literally fooling the system to think that it has become fertilized, whereas actually you are inserting a somatic cell nucleus. So that when you insert it into the uterus, with the right conditions, you might have to give the proper chemical or electrical stimuli, and then the zygote will be formed, the embryo will be formed, the fetus will be formed, and a clone of the human A will be formed. This is highly controversial, and this is just for example. No clone of a human has ever been formed or ever been allowed, but the clone of a sheep was formed, which was called Dolly, and that clone was formed, and it was in news. But the problem is that the clone did not survive for long. It contracted a lot of diseases, arthritis, because what happens? I am removing the somatic cell of this person A, and then I'm trying to grow the clone of this person A, but I'm not removing the cytoplasm of this person A, have I? No. I'm not removing the nuclear membrane. I'm not removing the mitochondrial DNA. So there are so many factors. It's not just genetics, but it is the cell microenvironment which dictates. So when fertilization happens between a sperm and an egg cell, the cytoplasm, everything is there. So a proper fertilization happens. When I just remove the nucleus, I'm concentrated on genetics and I'm neglecting the epigenetics. And because of epigenetic abnormalities, there will be a lot of diseases that the clone will develop over the course of time. Arthritis, heart diseases, and the clone won't be able to survive. That's why Dolly the sheep did not survive for long. It is only when we truly develop this technology of epigenetic engineering, not just genetic engineering, that we can go further in this study of cloning. And by the way, cloning is a familiar word. The technical word for it is somatic cell nuclear transfer. All right, so many things in such a short video. It's hard to get anywhere else. So do value it. Do subscribe to Beacon and like and share and give your valuable comments. And if you're really interested in biotechnology, I hope you've got the breadth and expanse. So whatever is there in biotechnology, more or less it has been covered in this video. So I hope you like it and keep following Beacon. Thank you. This is Raman.butta signing off. Thank you.